Thank you very much. It's now my pleasure to introduce my very good friend and collaborator, Ray Kurzweil. The short form of his biography is in your materials, but I think what might be more meaningful to you is for me to share more of a personal introduction. Ray and I recently realized that there are probably no two people that either of us know, and we're both fortunate to know hundreds of people, who have a higher correlation of interests, who have more interests in common with each other than the two of us have with each other. And that certainly makes my life very enjoyable, and I know it adds enjoyment to Ray's life as well. Just to give you a short summary, Ray is leading our research effort into a new type of cancer stem cell that is being done at MIT that, if successful, could result in a blockbuster cure for cancer, something that Barack Obama mentioned in his speech last night. Ray helped redesign our telecardiology algorithm that we use to monitor patients' P waves, QRS intervals throughout the country and help prevent heart attacks. He and his team, using their pattern recognition expertise, helped us re-engineer that algorithm, and it's now being used by thousands of people throughout the country. He's on the board of directors of United Therapeutics, so has helped us identify the best areas of activity. And I've also been very pleased to collaborate with him in the nutritional supplement area and even areas outside the immediate purview of technology. We are collaborators on a film version of his best-selling book, The Singularity is Near, that should be ready for release either toward the end of this year or thereabouts. And so we just have a tremendously wide range of activity. I really believe that his books are among the most prophetic in the best sense of the word, meaning a true vision of the near-term future that have ever been written by anybody. And Ray usually speaks to groups of hundreds and even thousands of people. So it's a really rare treat to have a private lecture by Ray to a group as small as this. And just heart goes out to you, little brothers. Thanks so much for being here.
Well, thank you for that. Thanks for that warm introduction. This has been a very gratifying friendship and relationship, multifaceted one, a personal relationship, and we have worked together, as Martine indicated, on many different projects. I'll tell you a little bit more about the cancer project. I've had the privilege of serving on the United Therapeutics board since it went public. That is actually a very unique company. It's the only biotechnology company I know that goes completely from basic scientific research to being a commercial success. Most biotech companies de-risk a project, take it up to in vitro tests, maybe animal tests, maybe they go to phase 1. But United Therapeutics is the leader in pulmonary hypertension treatments, having gone from basic science to a high level of ongoing commercial success, and it's now researching and developing drugs in many different areas, including cancer, as Martine mentioned. United Therapeutics exists in an alternative universe. It's a universe that doesn't have recessions, doesn't have stock market crashes, and it has relatively few, there are no failures. Failure is just success deferred, because Martine shares my philosophy that when you meet challenges, you just go around them, and ultimately you get there. Edison could have stopped when he was advised to after 1,000 filaments didn't work. So it's been a great privilege to work with you, and I look forward to many other collaborations. And biotechnology, by the way, is the foundation of what we're talking about today, because as you can see from the presentations, nanotechnology can only be done with a basic understanding of biology. And most of the presentations have been very intensive in terms of understanding the mechanisms of biology and how we can then go beyond them by extending them with our technology. And that's what human beings do. We go beyond our limitations. We didn't stay in the ground. We didn't stay on the planet. We have not stayed with the limitations of our biology. Very few of us would be here. I see a few young faces, but life expectancy was 23 1,000 years ago. So without the technological progress we've made, at best we'd all be senior citizens. And this is going to go into high gear. I want to talk about exponential growth. That's a key theme. And the reason that my projections sometimes seem daunting or radical to some observers is that they leave out the exponential nature of progress in information technology. And there's a good reason for that. Our intelligence is designed to make predictions. That's what intelligence does. And when we walked through the savannah 1,000 years ago, we saw something coming at us through the corner of our eye. We made a linear prediction about where that animal would be and what to do about it, and that worked quite well 1,000 years ago. And we have built-in predictors that are linear. That's our intuition. But our intuition is wrong when it comes to the progress of evolutionary processes. And I'll show you this applies both to biological evolution and technological evolution. And in fact, technological evolution emerges smoothly out of the biological evolution that led to the technology creating species. And that progresses in an exponential manner. And we can actually measure that. And it turns out if we can measure the underlying information properties of a technology, it progresses in an exponential manner. Depending on what we're measuring, the doubling time might be 2 years, 1 year, 15 months, 9 months, but there's an exponential progression. And that is not intuitive. So when people look out, they make linear predictions about where technology will be. And if I take 30 steps linearly, 1, 2, 3, 4, 5, I get to 30. If I take 30 steps exponentially, 2, 4, 8, 16, I get to 1 billion. It makes a huge difference. And this is not just a projection. I've been doing this for 30 years. I decided I would be an inventor when I was 5. 30 years ago, I realized the key to being successful as an inventor was timing. Most inventors fail not because they can't get their gadgets to work, but because the timing is wrong, the enabling factors aren't in place when they need to be. And we do some mentoring and early stage investing, and we get a lot of business plans. And I would say 90% of those teams would build exactly what they claim if given the resources, but 90% of those projects would still fail because the timing is wrong. And being an engineer, I started gathering a lot of data. This has taken on a life of its own. I now have a team of 10 people that gathers data in different areas where we can measure the information properties. And 30 years ago, I started to make mathematical models of how technology progresses in different areas. And really, to my surprise, I saw that it really follows extremely predictable trajectories. And the common wisdom is you can't predict the future. And that's true for specific projects, specific companies. There's one exception to that, which is United Therapeutics. But otherwise, you really don't know who's going to succeed or fail. Will Google stock be higher or lower than it is today? That's hard to predict. But if you ask me, what will the cost of a MIPS of computing be in 2012, or the cost of sequencing a base pair DNA in 2014, or the cost of a megabyte of brain data, or the spatial resolution of brain scanning in 2016, I can give you a figure, and it's likely to be accurate because my predictions have been accurate for quite a long time. In the early 80s, I saw the DARPANET doubling every year. It was only a few tens of thousands of nodes, so I predicted a World Wide Web connecting hundreds of millions of people emerging in the mid-1990s, and that was seen as ridiculous and was heavily criticized when the entire DoD budget could only fund a few thousand scientists on the DARPANET, but it came to pass right on schedule. I saw the chess supercomputers doubling in power every year, and that put the crossover where a computer would beat the world chess champion in 1998, and that was scoffed at in early 80s when the average chess player could beat the best machines, and Kasparov scoffed at it in 93 when he said, I've played the best chess machines in the world, and they're pathetic, brittle, predictable, but they soared past them in 97. And the Genome Project was controversial when it was announced in 1990, because mainstream scientists said we used our best equipment, our most advanced graduate students, and around the world we collected 1/10,000th of the genome. This is going to take centuries. And halfway through the project, the skeptics were confirmed, saying, I told you this wasn't going to work. I mean, here you are halfway through a 15-year project, and you finish 1% of the project. But that's right on schedule for an exponential progression, because you start out doubling little numbers, which might actually be sublinear. And if you double 1% 7 more times, you get 100%, and that's exactly what happened, and that's continued past the end of the Genome Project. So those of you who've read my book, Singularity is Near, you see I have numerous graphs in different areas of technology where you can measure the information properties. They follow amazingly predictable exponential progressions. I wanted to update them for this presentation, so you will see presentation graphs now that are updated to 2007. Singularity is Near is a 2005 book. The graphs are up through 2002. So we recently updated them to 2007. My staff asked me, aren't you a little nervous? I mean, maybe it didn't follow the graphs. It's not like we control this, and it's not a tabletop physics experiment. And I said, well, you know, maybe it won't come out right, but that'll probably reflect the problem with the data, not the actual progressions. But they came out right on the progression. It really never ceases to amaze me how predictable this is when these graphs are a measure of the innovation and the invention of millions of people, like all of you in this room, around the world, and they follow these amazingly predictable projections. Except that a lot of your presentations this morning, you're making progress faster than my predictions. My prediction is on size reduction, a factor of 100 in 3D volume. That's about 4.5 in linear dimensions per decade. And a lot of you, if you compare the presentations today to the ones a year ago, are faster than that. So slow down. I like to keep my graphs neat. It's hard to explain if we go off the graph, even if it's in a good direction. So we have time. It's very exciting to see the progress on the respirosites. I'll show you a brief video of that, but I've been talking about that for a long time and have funded some of our predecessors' work, and it's exciting to see that let's actually make progress on that. I don't have quite $1 quadrillion. I could give you half down. It's a down payment. Well, obviously, we need to make exponential progress, but that's exactly what we will do. So one of my main themes is that progress in information technology is exponential, and it's predictable. And moreover, it's not just Moore's law. People often call this Moore's law, and they say, well, how long will Moore's law last? Moore's law is just one example. It's just one paradigm, even within computation. It was not the first paradigm to bring exponential growth to computation. The exponential growth of computation started, I have data back to the 1890 American census, decades before Gordon Moore was even born. And it's very smooth, doubly exponential growth. Moore's law, the shrinking of component sizes on an integrated circuit, was the fifth, not the first paradigm to bring exponential growth to computing. And what happened, people say, well, you can't just take these exponentials and project them out indefinitely. Exponential growth comes to an end. You get 2 rabbits in Australia, and you get 4 8/16 rabbits, but finally they eat up all the foliage and the exponential growth stops. And indeed, all of these exponentials run out of resources. But what happens is as we see the end of the road for a particular paradigm, it creates research pressure to create the next paradigm. And the next paradigm is there generally in time. Sometimes if you have enough data, you can actually see a little ripple as the cascade of S-curves that makes up the exponential can be seen on these graphs. But this has actually happened now 5 times in computation. The first predictions for the end of Moore's law were 2002. Gordon Moore now says 2022. If you talk to Justin Ratner, who's the chief technology officer at Intel, he will tell you they already have the 6th paradigm working in their laboratories, which is 3-dimensional molecular self-organizing circuits. Those self-organizing electronic circuits will be part of nanobots to provide the computational resources that are needed, and they have early prototypes of these kinds of circuits working, and they're already actually, look at your cell phone, it has some early harbingers of three-dimensional circuits working in there. But it's not just computation that is subject to this, what I call law of accelerating returns. It's any technology where we can measure the information properties. And industries go, and areas of science and technology go through a transformation from a pre-information era to a post-information era. Now, health and medicine was not an information technology up until very recently. We didn't have the genome until 2003, and we didn't have means of manipulating it, and we couldn't simulate biological processes, et cetera. Now, we made progress. Life expectancy was 23. 1,000 years ago, it was 37 in 1800. There was no sanitation, no antibiotics. We didn't understand the germ theory of disease. Mozart and Schubert died in their 30s, and that was typical. So it was 48 in 1900. We've made progress, but that's been linear progress. Health and medicine has been hit or miss. We would just find something. Oh, here's something that lowers blood pressure. We don't really know why it works. There's no really good theory of operation. We can't design or simulate these drugs. Most of the drugs on the market today were done that way. It's called drug discovery, discovering something that happens. Like Viagra was a blood pressure medication. They asked people to send them back, and all the women sent them back. The men didn't send them back. They looked into why that was, and they made an accidental discovery. That's how drug development has been done. Now we have some new set of tools. We collected the genome, which, as I said, was a beautiful example of the law of accelerating returns. I'll show you how exquisitely smooth and predictable that progression has been in terms of the amount of genetic data we've collected, the cost of that genetic data, and this is also now true of other aspects of reverse engineering biology, like the proteome project, the connectome project to understand the human brain, and so on. We also have means now of actually changing what amounts to outdated software in our bodies. I mean, when's the last time you updated your software on your notebook or your phone? It's probably a few weeks ago. In fact, your phone now updates its software automatically without you even knowing it. We still have software running in our bodies. That's what our genes are. They are data programs. Okay, they don't work exactly like the sequential linear software on our PCs. They have three-dimensional properties. They interact with the three-dimensional properties of proteins, but they are linear sequences that give rise to linear sequences of amino acids that fold up in a predictable way, and we have, in fact, made progress on protein folding, at least in simulation. We can now actually simulate at least short sequences of amino acids and predict how they will fold up, and that's been a recent development on the latest supercomputers. But the software is outdated. It evolved thousands of years ago. The fat insulin receptor gene says, hold on to every calorie because the next hunting season may not work out so well. That made sense when that gene evolved. Actually, it's a very ancient gene that allows animals to roam around. Plants don't have a fat insulin receptor gene. But that, in today's world, which in most parts of the world are an era of abundance rather than scarcity, it's led to an epidemic of obesity. We'd like to turn that off, at least in the fat cells. You can't turn them off in the muscle cells. That would lead to a muscle wasting disease. We have a technology now, RNA interference, that can turn genes off. The fat insulin receptor gene was turned off in mice at the Johnson Diabetes Center. These animals ate ravenously and remained slim, and it wasn't a fake slimness. They didn't get diabetes, they didn't get heart disease. They lived 20% longer. They got most of the benefit of caloric restriction while doing the opposite. There are several pharmaceutical companies rushing to bring that idea to the human market. That's just one of the 23,000 genes we'd like to consider tinkering with. And RNA interference just started working a few years ago. It's another one of the prerequisites that has now made health and medicine an information technology. And as an example of the acceleration of progress, it was recognized with the Nobel Prize within a few years of it being shown to work. Usually in times past, that would take 20 years. Now it took 3 or 4 years. We have new means of gene therapy. They can add new genes. So one of the pioneering projects at United Therapeutics is to take cells out of the body, in this case lung cells, add a new gene in vitro. Now you can expect that it got done correctly, that there aren't other troublesome genetic errors. It doesn't trigger the immune system since you're doing it in vitro. That's been an issue that has plagued gene therapy in the past. Then that cell is replicated, and so it's inspected with a new type of microscope. That's another new development. It's replicated a millionfold. It's another new technology, injected back in the body. They're lung cells, so they end up back in the lungs. And this has cured pulmonary hypertension in animals. It's undergoing human trials. It's one example of many. There are about 1,000 drugs or processes in the development and testing pipeline using either RNA interference or new forms of gene therapy to basically update the outdated software that runs in our body.